over time, and hopefully people will keep zooming in as time goes. So I'm sure it's trouble. I see that Lori's nodding her head. She also had trouble. So with that, I will say it's really my pleasure to introduce Dr. Folami Ideraabdullah. Did I get it? I got it. Okay. I'm really thrilled that she's here. She received her bachelor's degree from Penn State University in biology with a focus on genetics. Following that, she obtained a Ph.D. from University of North Carolina, Chapel Hill, where she focused on mouse genetics, and then completed a postdoctoral fellowship at the University of Pennsylvania, focusing on genomic imprinting. Then she went back to University of North Carolina at Chapel Hill, where she joined the faculty in genetics and nutrition. And fairly recently, she was promoted to associate professor. So congratulations on that. She's a very successful researcher who is well funded by NIH and has many outstanding publications. She's also an outstanding mentor who has trained several postdoctoral fellows, PhD students, and undergraduates. A very active member of scientific societies, including the Society for the Study of Reproduction, the Environmental Mutagenesis and Genomic Society, and she currently co-directs the Development Disease Research Group at University of North Carolina, Chapel Hill. She's also won many honors. Two that I want to point out fairly recently. She was highlighted in the Society for the Study of Reproduction's blog as part of Women's History Month, and she's been listed several times now as one of the 100 inspiring Black scientists in America. So really wonderful achievements. And with that, I will turn over the seminar to Folami. Thank you.
Thank you so much, Jody, and thank you all for coming. I hope that I can, let me make sure I'm doing this right. I can present something that would be both interesting and hopefully useful to you and maybe spur some conversation. Can you all see my slides? Jody, can you see my slides?
Yes.
Oh, great. Great. So let me just turn on this laser pointer so I can use that. All right. So yeah, so it's my pleasure to be here. Thank you, Jody, for inviting me. I've known Jody for quite a few years now. I think we've danced at least one GRC, so we have connections that go back, and I really appreciate the opportunity to share some of our work. Most of it is published, but some of it's not, and I'm happy to chat about either component of it. I'm going to talk a little bit about our work where we're using different types of mouse models to understand or at least track interindividual effects of the environment on sperm DNA methylation. And I'm focusing on sperm DNA methylation in this talk, but we also obviously look at somatic cell methylation as well as different types of phenotypes, which I'll touch on, but not sort of go into too much detail of. And I hopefully can get through this talk because I made the rookie mistake of trying to show two different models because I wanted to show that we're doing this in kind of two different ways. So if I speed through some stuff, don't worry, I'm happy to still chat about it after the talk. So my lab primarily focuses on trying to understand genetic and epigenetic mechanisms of response to environment. By training, I am a geneticist, and I'm always interested in trying to understand heritability and what are the factors that are responsible for heritable phenotypes and long-term phenotypes. But I became interested in epigenetics, and these days you can't think a lot about epigenetics without thinking about what the developmental environment is doing to epigenetics. We look at this question from three different angles, or I would say maybe three different types of models, trying to understand, use the animal model and mouse models as a model of what's happening maybe in human developmental environments. And then we do a little bit of work directly with human tissues, specifically placental tissues. So we mostly focused on understanding the impact of diet and nutrition, the impacts of toxicant exposures, and then of course the impact of maternal health on short and long-term offspring outcomes. So how does this developmental environment contribute to these outcomes, and why do they persist? What is regulating their the phenotypic severity, for example? And then also, what are some things, hopefully down the line that we could do to prevent or treat these types of outcomes? We mainly look at growth and developmental phenotypes. We started recently to look at more metabolic endpoints, adiposity or fat accumulation, liver metabolism, glycemic regulation, those kinds of phenotypes. But a lot of the work that I did early on was really looking at reproductive endpoints, primarily in males. And so that's what I'm going to talk to you about today. And we think a lot about, what are the epigenetic mechanisms perturbed by these developmental environments? What kinds of epigenetic mechanisms are driving some of the disease outcomes we see? And then finally, how do genetic mechanisms really contribute to why individuals respond differently to these developmental environments? As I was telling someone earlier, I started my career with a very basic view of what was happening during development that really only involved genetics. And I think that we do have to think about genetics, epigenetics, and exogenous environments as they contribute to this developmental environment. And so we do a little bit of that. And so, as I said, I made a little bit of a rookie mistake of putting 2 models in, and hopefully I can get through all of it. But the first part of what I want to talk about, because I know there might be some people that are interested in it, and I know Jody's lab does some work on pesticide exposure, but this was a model that we started our questions with, and trying to understand the impact, really the role of genetic differences in response to pesticide exposure in our mouse models. So I don't have to tell you, but pesticide exposure is widespread. In the US alone, there's a really high level of pesticide use, and worldwide, obviously also high. Restrictions to pesticides obviously are present and do occur, but they really vary by compound and country. And so even when you get rid of pesticides in the US, they're still often used overseas, and some of those products are still imported. And so there's agricultural use to be concerned about, recreational use, meaning things like golf courses or parks, for example, and then personal use. A lot of people use pesticides in their own home sort of landscaping and garden care. There's lots of different routes of contamination that we know of: oral, dermal, vegetation, so things you eat or are exposed to through the skin. And then there's a lot of concern around pesticides in the groundwater, particularly for pesticides that have long-term lifespans. And so we became interested in pesticide exposure because of studies that have accumulated showing that pesticide exposure during pregnancy or during that developmental window has a long-term effect on the offspring. So in addition to well-known pregnancy complications, there's also some studies showing neonatal health effects. But as I said, we're really interested in potential long-term effects on child health. This is a short list of things that have been published in epidemiological studies. And so I just wanted to point out some of the key things that have been found. So the model we work with is vinclozolin. It is a fungicide. It has more than 15 metabolites, and it is a known endocrine disrupting chemical, actually one of the first sort of researched, or I would say published, endocrine disrupting chemicals, one of the first that was shown to have both anti-androgenic activity and anti-estrogenic activity, or estrogen agonistic activity, I should say. But it has been heavily restricted in the US, other than use on wine grapes. It's still used on turf grass. And then it is used on some imported fruits and vegetables, as well as ornamental flowers. It's not a pesticide that I would say is of high concern in the US, but it is still used in other countries. And there are other pesticides that have similar chemical structure to vinclozolin that could be of concern. as well as if our mechanisms are acting through these pathways, then there is concern of other types of compounds that have similar mechanisms. Lots again of different ways of exposure. I just wanted to show that, again, it was restricted in use in the US in 2005. And so you can see that use in the US has dropped off. However, as I mentioned, there are several other pesticides that have similar, at least from the carcinogenic component of the vinclozolin compound, have similar moiety to vinclozolin and are still widely used in the US in agriculture primarily. The other reason we were interested in vinclozolin, not just because of its role as an endocrine disruptor and a pesticide, but also because it was shown to have effects on male reproductive function in a number of different mammalian animal models. And at the time, I was studying imprinted genes and thinking about how the developmental environment contribute to genomic imprinting. And this is one of the papers that came out that got me interested in vinclozolin, because it was showing that vinclozolin exposure just during a very small window of pregnancy and gestation was able to alter DNA methylation marks at multiple imprinted loci and also alter sperm concentrations. At least for the epigenetic changes at the imprinted loci, was shown across multiple generations, which suggested some sort of persistent or more permanent effect, although the changes in sperm sperm concentration were only in one generation. But our interest was in using this as a model to try to understand how genetic differences contribute to differences in response to this type of environmental exposure during pregnancy. So at the time, I was a pretty pure epigeneticist and geneticist. We were actually looking at the H19IGF2 region, and we were generating genetic models that would help us hopefully understand how this imprinting control region, which is an epigenetically regulated region at this locus, try to understand how that imprinting control region regulated expression of the genes that are flanking it. And I won't go into the mechanism of imprinting, but happy to chat with you about that. And so these two genes are really important, particularly, I think everyone knows insulin is very important, but insulin-like growth factor, IGF-2 and H19, play a very important role in fetal and postnatal growth and also in glycemic homeostasis. And loss of imprinting at this locus or dysregulation of expression, imprinted expression of these genes is related to Beckwith-Wiedemann syndrome, particularly IGF-2 dysregulation is associated with overgrowth, glycemic dysfunction, and obesity in offspring in humans. And because we are really interested in this and the fact that the locus and the mechanism of imprinting is conserved between mouse and humans, we decided to make a number of mutations at this locus to try to understand how different things like CpG density contribute to the epigenetic mark and the regulation of gene expression at that locus. And so I spent quite a bit of time making different mutations at this locus to try to understand how this region is contributing to Beckwith-Wiedemann syndrome related phenotypes in the mouse. And what we found was really interesting, even though we changed, the length of this regulatory element, changed CpG density. For the most part, there was very little impact on methylation at the region, and there was very little impact on expression or offspring growth. And so that's when I did become interested in environmental exposures. And we decided to start with vinclozolin to try to understand whether these genetic changes are not alone causing some of these phenotypes, but maybe in combination with the environmental exposure causing these phenotypes. And so this is our treatment scheme. I'm not going to go too much into detail, except that we expose mom during pregnancy, during gestation, expose essentially the pups during gestation, and we expose them during a window of time where the sperm epigenome or the germline epigenome of males is being programmed or is being established. We treated them with either vehicle control or with vinclozolin. And then we looked at the effects sort of later in life postnatally. And as I mentioned, this is a period of time in the germline where the epigenome is undergoing developmental reprogramming. So in both male and female germ cells, E9.5 is probably somewhere around here where these epigenetic marks have been erased in early development and primordial germ cell development and now get reestablished. And so we're looking at how exposure during this very sensitive window of establishment of these epigenetic marks affects not just reproductive capabilities, but epigenetic marks. And we chose to do this looking at both vinclozolin, and we also looked at M2 because we wanted to ask whether anti-androgenic activity was driving these phenotypes. And so a lot of studies had already looked at vinclozolin exposure and showed that it affected DNA methylation marks across the germline, but they hadn't necessarily compared it to M2 to see whether this is anti-androgenic activity driven. And so that was our way of sort of asking that question. And then our primary question was whether mutations at that imprinting control region would increase sensitivity to these changes. So is there a gene by epigenome interaction or gene by environment interaction that's causing these epigenetic changes? And so we compared wild type animals to what I'm calling ICR mutant animals, one of our ICR mutations that altered 8 base pairs at this imprinting control region. And so all the data I'm going to show from this particular project is comparing those two mutants. And so the first thing we looked at was obviously fertility and fecundity, and we do that through looking at litter size of mice and then looking at things like litter survival to weaning. And so even though we showed that the mutation alone did have an effect on survival to weaning, we didn't see a significant effect of either vinclozolin or M2 on fertility or fecundity when we looked at it. We also didn't see an immediate effect of vinclozolin on things like body weight, which we use as a measure of development. You can see a little bit of an increase in body weight in our vinclozolin treated group, no effect in the M2 group here. These are our wild type animals, maybe a little bit of a similar trend in our mutant animals, but none of it was statistically significant. The only thing that was significant was a slight increase in our M2 animals in neonatal. And so far, basically, not much happening. We did take some other measures. This is probably the one that was most changed, but so far, not much was happening at the neonatal stages. However, when we looked at these animals later in life at weaning, keeping in mind they were only treated for 10 days during gestation, we did see that there was an increase in body weight only significant in our mutant animals, and an increase in fat mass in our mutant animals, percent fat mass. So after adjustment for body weight, there was still increased adiposity in these animals. And we saw the increased adiposity in both male and female offspring that I'm showing here. No effect of M2, which was the more anti-androgenic compound, just with vinclozolin. We also looked at these same effects in adulthood. And again, we still saw a very strong effect on offspring body weight caused by vinclozolin. There was still a trend of increased fat mass, but it wasn't statistically significant in adulthood. And so we moved on then to ask about a couple of reproductive measures. So vinclozolin as an anti-androgenic compound has been shown previously to cause decreased anogenital distance. And so we wanted to see whether we're seeing that, and of course to test whether our M2 treatment is having any effect on anti-androgenic related effects. And so as we expected, the M2 did cause a decrease in anogenital distance only in our mutant animals. There was no significant effect of vinclozolin alone in either our mutant or our wild type animals. And there was no effect at all on sperm count in these animals. So very minimal effect on these sort of reproductive developmental measures. We also started to look at methylation. So one of the first reasons we asked this question was to try to understand whether this genetic mutation would increase susceptibility to methylation at this locus. These are my 2 genes that I mentioned before, and then this is the imprinting control region that we've mutated. And when we look at somatic tissues, for example, fat that might be related to the increased adiposity, we didn't see any significant change in fat mass, in DNA methylation at this locus for either of our treatments. And so essentially in adults, which is where we're measuring these things, we did not see an epigenetic or increased susceptibility to epigenetic changes at the ICR in somatic cells overall. But when we looked at sperm methylation, we did see a slight decrease in DNA methylation in sperm, really small decrease, but we did reproduce it in a second set of animals. And so it's a really small decrease at this region. It's only in our mutant animals again, and we did not see the same phenotype in our M2 animals. So this was very specific to our vinclozolin untreated group. And then finally, we wanted to ask specifically about this mutation and whether it was being transmitted through the maternal or the paternal germline, whether that made a difference. And so what we did was we compared wild type animals. This is a completely separate set of treatment studies that we did secondarily. We compared wild type animals to either animals that inherited the mutation through their mom or animals that inherited the mutation through their dad. And we were able to show that we were only able to see that increase in body weight and decrease in sperm methylation at the ICR when it was maternally transmitted. So this is a maternal specific gene by environment interaction that is not observed from the paternal germline. The other thing we did was we wanted to separate maternal versus offspring genetic susceptibility. And there's many ways you can do this. I know sometimes you can do cross fostering. Well, we decided to do it in a little bit more complicated way. So basically we took heterozygous moms who will have either wild type or mutant offspring, and we transmitted it that way through the mother or the father. And what we found was that, again, only seeing the effect through the mom, and both wild type and mutant offspring of these moms were affected similarly for both the body weight and the sperm methylation change. And so that tells us that it's really the maternal genotype that's driving this response, and something that we would have completely missed if we weren't able to distinguish those things. So hopefully I didn't go through that too fast, but we had a couple of conclusions just from this sort of simple study. And the first is that the genotype at this locus does increase susceptibility to in utero vinclozolin exposure. As I said, this is an imprinted locus, but it has previously been shown to play a role in metabolism and growth. So we weren't completely surprised that we saw increased body weight and adiposity. We were surprised that we didn't see any change in ICR methylation in adult somatic tissues. But we have shown in other studies that it may not be a methylation mark. I've talked to other people, it could possibly be a different type of epigenetic mark that's driving these changes. And we only looked at methylation. We did see a subtle loss of methylation at the ICR and sperm, again, in these mutant mice only. Probably not enough to drive any strong phenotype in the sperm or in the subsequent generations. But it was there and it was consistently found. And so I think in total conclusion, it's that the parental and not the pup genotype is driving susceptibility to some of these phenotypes. So it complicates things a little bit when you're thinking about this from a human perspective, but it also helps us understand that if we're thinking about this from the perspective of, can we use these types of mutations to predict susceptibility of the offspring, it actually makes it a little bit easier if we're thinking about parental genotype and not pup genotype. And I'm not saying this is true of all genetic susceptibility to pesticides. This is just one model that's showing pretty clearly that it's parental genotype and specifically maternal genotype. What's the mechanism by which this is happening? I mean, we can definitely discuss that. We don't know the answer to that. I think it could potentially be operating through some sort of placental phenotype, which we haven't tested yet. And so that's probably one of the major study limitations that we're focused on trying to address. We only looked at effects in adulthood, and we really need to test earlier stages of development and think about whether this is a fetal versus an adult onset effect epigenetically. We know the phenotype is more of a post-weaning adult onset, but in terms of when those molecular changes occur is what we need to test next. So I'm going to move into part 2 of my talk, which is focused on a lot of the work we've been doing more recently, which is looking at the effect of vitamin D deficiency on in the developmental environment on some similar endpoints as I showed you for the pesticide exposure model. Vitamin D is a micronutrient, a very important micronutrient that plays a very important role in development and has been linked to a number of many different types of diseases. I always say vitamin D is linked to everything under the sun. with stronger evidence for some versus others. But it is a micronutrient that you can get through diet or sunlight. And we were really interested in it because it does have hormone-like activity. And so vitamin D operates as a steroid hormone, and it binds to a vitamin D receptor, which can directly bind to the genome and either activate or repress gene expression. There are many vitamin D receptor elements or the binding sites for these this complex across the genome. They differ by cell type, they differ by developmental stage. And so vitamin D has that potential to directly regulate gene expression. And then there's also some evidence that vitamin D has some non-genomic signaling pathway effects as well. And so we're interested because obviously we're looking at DNA methylation, which is a genomic mark that's attached to the DNA sequence, and wanting to understand how vitamin D could potentially play a role in that. We're also interested because vitamin D is pretty prevalent during pregnancy, and there's not really, there's some data showing associations between vitamin D during pregnancy and childhood outcomes, which I'll show you in a second. But it's not really clear the extent to which this deficiency could impact offspring or multiple generations of offspring. And so there, again, similar to our pesticide model, we chose vitamin D because it does show increased risk of adverse effects at birth, but also increased risk of adverse effects later on in life. The other reason is that there's quite a bit of evidence, mostly in ex vivo studies, so in cell cultures, showing or tissue-related cultures showing that vitamin D plays a role in or regulates histone demethylases, and that vitamin D also can regulate the recruitment of various epigenetic complexes, including histone methyltransferases, histone acetyltransferases, and so on, as well as chromatin remodelers. We chose to stick with looking at DNA methylation just because it hadn't been measured before. It was a lot easier to measure across development. But we are very interested in going back now and thinking about how histone modifications might be affected as well. This is our model that we used. It's not our model. We didn't develop it ourselves, but it's a model that has been used in the field of vitamin D deficiency, comparing basically a sufficient level of vitamin D to a diet lacking vitamin D. And we treated these mice, moms, with this diet, both before conception to make them deficient at conception, and then we treated them throughout gestation and lactation. The pups were put on a vitamin D sufficient diet. So again, only wanting to understand what's happening during development and this developmental environment and how it contributes to long-term outcomes. For this model, we're actually using the collaborative cross, and that's because we're really interested in naturally occurring differences to better mimic a human population, right? To my knowledge, most people don't carry a full knockout of any gene. Some people do, but most people do not. It's not generally tolerated that important genes are knocked out, and people pass those on don't pass those on pretty regularly. And so we chose the collaborative cross because it is a population of mice with a high level of genetic differences, a high level of genetic divergence among the different strains. And in fact, it captures about 90% of the genetic variation among laboratory mice. So pretty high level, and also being able to look at, what is contributing from different mouse strains. And the other reason we're really interested in these strains is because they're inbred. And so we can do some biological replicate work for the gene by environment interaction that we're interested in studying. The first thing we found when we started looking at these lines, which I wasn't expecting to this degree, but why not? We found that the strains really differed in how they, I guess, metabolize vitamin D, or maybe even absorb vitamin D. So this is their circulating plasma vitamin D status, which is commonly used in humans to say whether someone's deficient or sufficient. And we saw that each one of these strains measured separately showed a difference in their vitamin D status, regardless of whether they were on the control diet or the low vitamin D diet. And one of the things I'm not showing you here, I should have added a figure, but there was also a difference in how they responded to the low vitamin D diet. So how depleted these mice became after being on the diet. Some strains maintained their vitamin D status longer, some strains became depleted very, very quickly. And so when we're looking at how these strains differ in their response to vitamin D, I think a component or a big component of that is also looking at how these strains differ in their vitamin D status and vitamin D metabolism. The first thing we showed with these animals, because we wanted to ask about growth and adiposity, was we looked at body weight and fat mass, and again, showed that, as has been shown for epidemiological studies, some of the strains did show an increase in body weight and fat mass later in life. However, there were many strains that didn't show any response or very little response in the other direction even. And so we've kind of labeled these as strains that are resistant and strains that are susceptible. And what we're working on now, and I'll show you a little bit more of, is kind of trying to compare susceptible strains to resistant strains to understand the mechanism by which strains are susceptible versus resistant, with both being sort of equal interest to my lab. So I'm going to show you a little bit of data from two different strains that we've been working on for a while. This is their plasma vitamin D status. This is an example where they had very similar vitamin D status, but when they were put on a vitamin D deficient diet, which are the white bars, this strain maintained higher levels of vitamin D compared to this strain. This is the strain that showed increased adiposity, whereas this is the strain that showed unchanged adiposity. The first thing we looked at in these mice, or one of the first things I should say, is reproductive measures, very simple measures. I believe we also looked at anogenital distance. There was no change. There was no change in sperm count for either of the lines that we looked at. There was this weird increase in testes weight in the susceptible line, and maybe a trend of increase in the resistant line that was not statistically significant. And so in the first round of sort of preliminary studies, we looked again at imprinted genes. And in this case, we looked at not just the H19 IGF2 region, but we looked at other genes, both maternally and paternally methylated imprinted genes, to try to understand whether there was sort of widespread dysregulation of methylation during development caused by vitamin D deficiency, and whether we could track that into adulthood. And so what we found when we look at adult animals from these vitamin D deficient, developmental vitamin D deficient models, and compared across these two strains, susceptible versus resistant strain, each one of these blocks is a different locus and methylation levels. The bar indicates the average methylation level. We saw that there were several regions that were differentially methylated. They were all loss of methylation, and they were all in the susceptible strain, whereas the resistant strain was not significantly altered. This is liver. We also looked at sperm from these matched animals and showed something very similar. Different regions affected in sperm, but again, very similar decrease in DNA methylation. And in this case, it looked like both the resistant line is significantly different. The susceptible line was trending in that direction, but not significant. And so it suggests that the effect that it's having on the germline and the effect it's having on the soma are somewhat different. But in both cases, it's loss of methylation. And so we decided to sort of pursue that question a little bit further and ask what's happening genome-wide. So is this an epigenetic landscape or epigenetic profile, a DNA methylome issue, or is this restricted to imprinted genes? This was a collaboration with Corey Brower and Rod Garibay at the University of North Carolina, Charlotte, when they were both there. And we used bisulfite targeted by, well, it's not really targeted bisulfite sequencing, but it's called targeted bisulfite sequencing. It's a bait sequencing library, which I'm happy to talk to you about. Agilent makes this kit. And it works, they have a kit for humans and mouse, so you can do a comparison. But at the time, it was the only way to really enrich for epigenetically regulated regions without having to do whole genome bisulfite sequencing, and the only way to really pull up CPG islands, as well as lots of other types of epigenetic regulatory regions, as opposed to just promoter regions, in a somewhat unbiased, non-sequence biased manner. We initially did the study just looking at sperm from our susceptible mice. This is just showing the coverage, that we had coverage across the genome. But what was really interesting when we look at the sperm methylation patterns across the genome, showing each chromosome, each bar is representing methylation of each chromosome. The red color is indicating loss of methylation, regions that have loss of methylation. The blue are regions that have gain of methylation. We found that most of the regions, first of all, there were differentially methylated regions across the genome, but most of them were loss of methylation, approximately 70%. And about 35% of those changes were due to a more than 10% change in methylation. The other thing that was interesting here is that it seemed like these sperm were really exhibiting loss of the hypermethylated regions. So most of the regions in sperm are hypermethylated. And so what we were seeing was a loss of methylation at those hypermethylated regions. And also when we compared our control samples to our vitamin D deficient samples, their methylation profiles were very distinct in terms of how they responded to this treatment. And that was true for changes that were loss of methylation and changes that were gain of methylation. And then finally, the other thing that we looked at, because we wanted to understand why these regions, or what is so special about these regions that are changing, we've asked a number of questions. I'm not showing them all here, but the simplest way we asked this was whether these regions are sensitive based on their baseline methylation state. And what I'm showing here is the difference in the extent of methylation change, or the effect size. So more than 20% difference in methylation, less than 10% difference in methylation. And the y-axis is showing the baseline methylation state. And these two panels are just different ways of viewing the data, separated by gain of methylation sites and loss of methylation sites. And what we found was that most of our highly differentially methylated regions or sites were actually in partially methylated regions. And so we think that it could mean that these partially methylated regions in sperm are more sensitive to epigenetic dysregulation, but that's a very, very broad hypothesis, and we don't necessarily have a mechanism as to why they would be more sensitive or not. We did find that most of the epimutations we found were in genic regions, so that was interesting as well. And we look at what kinds of pathways were affected. We found that most of these were developmental processes. Most notably, the cadherin and Wnt signaling pathways were enriched in our gene set. We did go in and validate many of these changes using pyrosequencing, so a second method, and most of them were validated as differentially methylated, and confirmed that there was differential methylation at many developmental gene, as well as many master epigenetic regulators, such as some of the histone demethylases, DNA hydroxymethylation enzymes as well. And so what we're looking into now is how those changes could potentially contribute to effects in the next generation, for example. This is really simple. We were just trying to test, one of the things we see a lot in environmental studies is a lot of heterogeneity in how the offspring respond. And so we compared basically offspring that responded higher at a high level to offspring that had very minimal response. And again, these are all genetically identical, but this is just some heterogeneity within the response of this line. And we were able to show that the methylation changes we found were primarily in the responders and not in the non-responders, which is what we expected, but it was nice to sort of show that not only were these guys not responding phenotypically, but they're not responding epigenetically either. All right, so some of the things that we have still going on is thinking about how these germline changes contribute to effects in the next generation. We're not doing a huge amount with that right now, but it is something that's always sort of on the back of our mind as to why these changes are relevant. And so we did do a very, very simple mating study to generate a second generation of animals. So we mated these males from the line that showed epigenetic changes in the sperm to unexposed females and looked at the second generation. No changes in fertility or fecundity again. but we did see changes in neonatal body weight. So neonatal growth is probably affected here. The effect did not persist into the second generation adults, only in the neonates. And we kind of saw going from postnatal day 4 to postnatal day 9 that the effects were attenuated over time. And probably why we don't see those changes in adults. But it is saying that the effects or the exposure of these males during development to that vitamin D deficient environment does affect their offspring, and it is going through the male germline. The other thing that was kind of interesting here is that when you compare this offspring from the susceptible versus the resistant line, and we're looking at female versus male offspring, we did see that there was a difference in how the second generation offspring responded. So it's not as apparent in the females, but in the males, you can really see that the susceptible line also showed an increase in body weight or growth. And the resistant line showed now a statistically significant decrease in body weight or growth. And so that gene by environment interaction that we see in the first generation is also affecting the second generation. What was interesting, but again, not super surprising, is that the methylation changes we saw in some of the key developmental and epigenetic regulators in the sperm were not directly inherited to the offspring of these males, at least the offspring that we tested. So we looked at neonatal liver from these offspring and don't see the same epigenetic changes in that second generation. And then here I was just looking at epigenetic changes at this one locus that we showed before, GRAB10, just showing that that was differentially methylated in the second generation of offspring as it was differentially methylated in the first generation. It was line specific again, with the susceptible line 1 being affected, and it was negatively correlated with the body weight phenotype that we see. So GRAB10 does play a role in adiposity and lipogenesis. So that's one area that could be of interest as we move further to see whether that's something that could be contributing to this phenotype. The other thing that we're working on right now is really comparing our susceptible and resistant lines at the level of the epigenetic response that we see and phenotypic response. But what I'm showing here is just at the level of the epigenetic response. So this is genome-wide DNA methylation, showing that our susceptible lines for both liver and sperm are more responsive, I guess, in terms of epigenetic perturbation compared to our resistant lines. And when we look at this from a profile level, what's actually pretty cool is that you can see really clearly, as expected, the samples cluster by cell type. Also as expected, because we showed this initially with the first set of sperm data, the samples do cluster by diet. But what was really interesting as well is that the samples also clustered by genetic line. So even within the vitamin D deficient treatment group, they cluster really distinctly by the susceptible versus the resistant line. And so these mice are responding differently at the level of their phenotype, but also at the level of the genetic perturbation that we're seeing in the germline, as well as in their somatic tissues. And what was kind of interesting even about the somatic tissues here, that I won't go into too much detail, is that these animals actually differed in both their baseline and response epigenetic profile based on their genetic lines. And that's something that we kind of touched on when we compared when we were looking at the initial study to see whether the baseline methylation would contribute to the extent of epigenetic perturbation in response to the treatment. All right, so in part 2, our conclusions of this model so far is that, again, genetic background influence susceptibility to vitamin D deficiency. I think this is pretty strong evidence of both influences in the susceptibility to the phenotypes, as well as influence on the susceptibility to epigenetic changes. Despite the genome-wide sort of loss of methylation in sperm, we didn't see a change in fertility. So that means that these sperm are likely not affecting the ability to fertilize the oocyte. But we do see effects on the second generation phenotypes. And that's what we showed next, which was that the VDD effects were transmitted through the male germline. So far, we've only looked at neonatal and developmental growth effects. that those genetic background effects that we saw in the first generation, we also see genetic background effects in the second generation pass through the male germline. And those effects that we see in the sire germline were not detectable in the neonatal soma. So at least at the level of the DNA methylation change, it's not a direct inheritance into the neonatal soma. We think it could be affecting development as a whole. And so again, one of the limitations of the study that we're now going back and doing is looking at earlier stages of development and looking at both embryonic and extraembryonic tissues as well. So the broader implications of these findings, because we really did start these studies and do them from the perspective of understanding human disease and how this would play out in a human population, is that when we're doing environmental studies, I think genetic background and genotype is indeed an important driver of the offspring response to the developmental environment. And any things that we can do to incorporate that into our study is a good idea. And especially because if we had not incorporated that into our study in the mouse, which is pretty easy to do in the mouse, but not as easy to do in humans, we probably would have masked these phenotypes if we were combining all the data from these genetically heterogeneous populations. And so another reason to sort of think about how we can incorporate genetic background or genetic ancestry even into some of these kinds of studies in human populations. And then finally, the fact that we saw that strain-specific multi-generational effect suggests that these gene by diet interaction effects are potentially happening over multiple generations. And it makes for an interesting concept as a geneticist to think about how these effects are heritable, because it doesn't immediately make sense. But they are, and I think thinking about how that could happen and the mechanism by which that could happen will help us understand, multi-generational effects of these developmental environments and how to both detect them and potentially prevent them or treat them. And so finally, hopefully I did not go too fast, but I want to acknowledge the people in my lab who are really the ones who do all the hard work. Megan is a postdoc in the lab. She kind of just started with me in the last about a year. She's been with my lab, and she's focused on the vitamin D deficiency models along with Liz. And then Asa and Amy are two undergrads in the lab that are also working with us on those models. Epigenetic was instrumental really in a lot of the pesticide exposure models. He was a technician in the lab and a lab manager with me for a long time. Now he's at Baylor doing his Ph.D. Was very sad to leave him, and I have to find someone else who wants to work on that study. Jing Shui is a postdoc who actually spearheaded this and started those vitamin D deficiency studies in my lab, and part of it was really her idea. So I give her all the credit for sort of getting me interested and excited in vitamin D. And all of the other people that contributed to these studies have just been instrumental, whether they're here now or have left now. My collaborators, I have a lot of different people who have either physically collaborated, meaning they did some of the experiments, or they've also sort of collaborated through, providing advice and information about either the genetic models we're working with or the environmental models. So for example, Mike Love, who worked with us to try to understand how to run the appropriate stats on these kinds of studies. As I mentioned, Corey and Rod, who helped us with the bioinformatics. Will, Lisa, and Fernando, who have a lot of experience with the collaborative cross mice before I started this project, and just provided a lot of information. Actually, the mice we're using were from a larger study that was Will and Lisa's effort to look at the effect of the developmental environment on behavioral health and neurological development. My collaborators at the University of Missouri, who have been instrumental in just helping me work through the vitamin D deficiency and the nutrition component of that study, as well as our collaborators who've really helped me think a lot more about vitamin D metabolism and actually test vitamin D metabolism. We have a collaboration with Bruce Hammock, where he's able to measure all of the different key vitamin D metabolites, which is something we're working on right now. to try to understand how genetic differences in vitamin D metabolism might be contributing to some of these offspring phenotypes. And then I didn't get to talk about our placenta studies, but I have a number of different collaborators who we're working with to ask those questions. Obviously, our funding mechanisms are very important. We can't do this type of research without funding because it is expensive and sort of a lot of effort. And I like to thank you all for listening. I'm happy to take any questions, and I will stop there.
Thank you so much for such an outstanding seminar. On behalf of everybody here, thank you very much. I like to say that this is now open for questions. People can feel free to unmute themselves and ask their question, or if you would rather, you could put it in the chat. So Prabhu, please go ahead.
Hi, Folami, that's a beautiful, beautiful talk. Really enjoyed it, the epigenetics aspect of it. I have a couple of questions. You know, that maternally transmitted phenotype is very, very interesting. Remind me, is that mutation within the IGF H19 locus?
Yes, yeah, it's a mutation that's at the imprinting control region. And that mutation, what it does is it replaces 8 CPGs or sites of methylation at the ICR. So it doesn't make a huge impact on the imprinting control region size, but it does reduce the CPG density. And there have been a number of studies that have come out recently that suggest that the CPG density is very important for maintaining proper epigenetic regulation at some of these types of loci. So yeah, so it is a mutation at the ICR.
But was that affecting the way the imprint is put in the maternal genome rather than in the paternal genome, right? It has to do with something, has to do with that.
Yeah, so that exactly. So, and I didn't go through that because I didn't want to make it more complicated than it already was. But yeah, so on the maternal allele, that locus is unmethylated. And on the paternal allele, that locus is methylated. And so it has a role on both the maternal and the paternal allele. And that's why we transmitted it in both ways to see if there would be a different phenotype or a similar phenotype when we transmit it both ways. But we only see the effect on the maternal transmission of that allele. We don't see it with the paternal.
Okay. And I have a second question about the second part, where you showed all those genome-wide methylation changes in your F1, I'm sorry, yeah, in the F1 males. And then you went ahead and mated them and they gave normal offspring. Did you by any chance have an opportunity to look at the sperm methylome of those G2s?
We haven't looked at the sperm, the genome-wide methylation of the second generation. What's kind of interesting that we did, and this is a 2016 paper in Clinical Epigenetics that I'm happy to send you, but when we did the initial study, we just looked at those imprinting control regions, I think six different imprinting control regions. And what we found was that in the first and second generation, the somatic and somatic, we used liver to represent our somatic tissue. The epigenetic changes we saw at the ICRs and the somatic tissues were similar between the G1 and the G2. And then the epigenetic changes we saw in the male germline, the sperm, were similar between the G1 and G2, but not between each other, right? And so the soma and the germline are somehow differentially disrupted in this way. We didn't go back yet and look genome wide. I think that would be a really, really cool thing to do. We started with, the comparison of the two strains, but that is one thing that we could do because we do have adult offspring from these. We just didn't pursue it as an initial question, but yeah, I don't know if it will be consistent across the genome as we saw at the few imprinted regions that we looked at, but it could be really interesting.
Thank you.
Yeah.
Laurie, go ahead.
Fantastic talk. Thank you so much for presenting for us. I have a couple of questions, but the one I'm most interested in hearing your answer to is about the vitamin D part. I thought that was so really important studies. And I'm wondering if you think the vitamin D deficiency directly, so if vitamin D directly affects the methylation or the offspring, or if you think that that's somehow affecting mom's physiology or placenta that then transmits the change to the offspring?
You know, it's a really good question. I don't know the answer because we haven't run the tests, and I don't believe anyone has run that test yet. I think that, let me think about this. So I think that there is potential for either, probably equal potential for either. We haven't seen, and again, there's an exhaustive list of things that we could test in terms of mom's physiology, but what we've seen so far is mostly liver metabolism differences in the moms. That's what we've seen and published so far. We haven't seen anything that would indicate, so for example, vitamin D deficiency is associated with gestational diabetes in humans. There have been some studies showing links. We haven't seen any evidence of gestational diabetes, either from mostly just looking at fasting glucose and glucose tolerance tests. But we haven't exhausted the list. So I would say at this stage, it could be equally likely a maternal, like a direct versus an indirect effect. One thing that we've thought about, yeah, but that wouldn't work either. Some people have suggested trying to do things like embryo transfer to sort of distinguish between the maternal and the fetal effect, which can get very complicated very fast in these strains because they're a little bit more difficult to work with. Yeah, we recently ran a test run to see if we could reproduce the effect in like C57 Black 6, which would be an easier strain to do that kind of, experimental analysis in. And the mice did not tolerate the diet very well at all. We had a lot of litter loss, which was interesting. So we're going back to look at why that happened. But yeah, I think it's equally likely. I don't have any data so far that we have that could distinguish between a direct versus an indirect effect.
Thank you.
Mm-hmm. You're welcome. Thanks for the question. It's a good question.
I think we have time for one more question. Diego, go ahead.
Wonderful talk. Like I said, this was amazing to just hear and, from firsthand. So my question was, in terms of the offspring, their vitamin D status, I assume that was normal from just having the mom on low vitamin D or no vitamin D. But in terms of their vitamin D receptor location, is it binding to the same regions? And even in somatic tissues like the liver, is it having the same distribution?
Yeah, so we had the same question. We didn't look at the offspring. We did look to see, the way we asked the question was kind of indirect. We asked whether the epigenetically dysregulated regions were enriched for vitamin D receptor element regions, and we didn't find a correlation there at all, or any enrichment, I should say. So we don't know the answer yet. And part of that was what we talked about a little bit earlier when we met, which was, the thing that I think we have to answer first is the timing of when these molecular changes are being perturbed. And once we have that, then we can ask those questions. Because I think in adulthood, my feeling is that that's well beyond whatever happened during development. And we're not going to see that even an indirect relationship between the vitamin D receptor binding and those response elements. So we need to look at it embryonically, or at least at the stage when we think and know that this perturbation is happening. And that's what we're working on right now, is thinking about what is the timing of when this would be occurring. So we have plans to look at several embryonic stages and early postnatal stages to see whether we can narrow that window down. But yeah, that would be sort of the next step is, and that goes along with Lori's question, which is this a direct effect of vitamin D binding aberrantly or not binding where it's supposed to bind, because it's not present, right, to bind the vitamin D receptor, or is this something else happening? Yeah. Good question.
I think that's all the time we have for questions. I would just like to say thank you so much. Really fantastic talk. Great work. So thank you. And I hope I get to see you in person sometime soon. And hopefully in the future, we can actually get you here for a physical visit would be great.
Oh, that would be fun. It was great meeting all of you. And I think I have a couple more meetings, but yeah, thank you all for coming. I appreciate it.
Thank you so much. See you soon.
Thank you, Jody. Bye-bye.